We are presenting the connection between MSH2 and HNPCC. I'm Lauren Dones.
I'm Naomi Akinpalu.
And I'm Chloe Press.
So to begin with, I want to talk about DNA mismatch repair. So the primary function of DNA mismatch repair is to maintain and preserve the genetic material. And to do so, it corrects mismatches that are made during the processes of replication, recombination, or mismatches that are made as a result of DNA damage. Because it's a vital pathway that prevents genetic mutations, which we all know can be very problematic. And within this is a key component called MSH2, and it functions alongside MSH3 and MSH6. So if the function of MSH2 is impaired, it can lead to very bad consequences, which we're going to talk about next. So the cause of hereditary non-polyposis colorectal cancer, also known as HNPCC, is linked to defects in genes that code for DNA mismatch repair. So like I just said, if the MSH2 is impaired, MMR cannot properly function as it's supposed to, and it can't run as efficiently as it would if MSH2 was working. So the mutations are then able to form at an increased rate and accumulate with no form of restraint. And this accumulation can lead to cancer, and in this case, HNPCC. The research was conducted to test if a particular single nucleotide polymorphism, also known as a SNP, and in this case, we were testing the SNP G704S, and we were trying to see if it affects the mutation rate. And we hypothesized that yes, this SNP will cause the mutation rate to increase. Therefore, we ultimately proposed that this SNP may correlate with a higher likelihood of HNPCC in humans.
So now I'm just going to talk a little bit about the methods, and there are three major methods that we performed in order to gain perspective surrounding our specific aim. And these include plasmid extraction and digestion, PCR and digestion, and yeast transformation in the FOA assay. So just to go into a little bit more depth about each of these, the main objective of the plasmid extraction and digestion. So for first, the plasmid extraction is to extract the DNA that is extract the DNA that's grown in E. coli bacterial culture, and then two, to identify the vectors that contain MSH2 gene and those that do not. So the first step was to isolate the plasmid DNA, and what we did to do this was first take the bacterial cells and put them in a microcentrifuge tube. Then we added a lysis buffer, which broke open the cells by dismantling the cell and nuclear membranes to let the DNA be free. Then we neutralized this lysis buffer so that it does not destroy the DNA. Then we bind and wash it and elute it so that we have a purified DNA sample for each of the strains that we use. So, and then finally, we saw how much DNA was concentrated in each of the samples by using a nanodrop. The next part of it was the digestion of the plasmid, and this was to see which of the identity of each of the plasmids. And to identify this, we used particular restriction enzymes that recognize a specific base set of bases and then cuts out a target gene. And by seeing how much is cut out of each plasmid, we are able to see which plasmid contains MSH2 or not. The empty vector will have a very small portion cut out of it, and then the plasmids that have MSH2, both the wild type and the one with the SNP, will have a larger small segment cut out of it. And this will identify whether it is carrying the MSH2 or not. So the next method is PCR and digestion. So after cutting out, the main goal of this procedure is to, one, amplify a specific range of DNA that contains the MSH2 gene plasmid. And to do this, you place the DNA along with primers, Taq polymerase, and free nucleotides into a centrifuge tube, and then cycle it through different heat or temperatures. First to denature it, so the denaturation of the DNA happens at around 94 to 96 degrees Celsius, and this acts to bind the two strands of DNA so that primers can anneal to them, and then the DNA strands can be elongated with the free base pairs. So then after the PCR, you have the digestion, and this digestion is to identify which plasmid has the SNP. So to the first one identified which has the MSH2 gene, and then the second digestion identifies whether or not the plasmid has the wild type MSH2 or has the MSH2 that has the particular SNP that we are studying. So the last method is the yeast transformation in the FOA assay. So the yeast transformation is to transform the yeast with these particular plasmids that either do not have MSH2, have wild type MSH2, or have the MSH2 with the particular SNP. And what we did to transform the yeast is place the yeast in with the particular vector, and then heat shock them to open up their cellular membranes, and then bring their temperature down rapidly so that they uptake the plasmid. And after they uptake the plasmid, we'll plate them on media containing FOA. And since the plasmid has a particular base sequence that is easily mutated, we can study whether or not the MSH2 gene that is injected into yeast that are mutated to not have MSH2, if this MSH2 that is injected with these plasmids is working or not. So the way that this is done is the particular gene that has a high mutation rate codes for turning FOA, which is in the media, into a toxin. And if the gene is working and does not have any mutations in it, then it will turn FOA into this toxin and kill the cells. But if it does not have a functional MSH2, these cells will not integrate the FOA into their DNA and will not die. So if there's growth on the plates that have FOA, then the subsequent mutation rate is high. And if there's no growth, then the subsequent mutation rate is low.
Okay, so here we have the result of the first method, which was plasmid extraction and digestion. Following the plasmid extraction and digestion, the DNA was analyzed through gel electrophoresis. The empty vector was expected to produce one band, while the plasmid with the MSH2 genes were expected to produce two bands. Here we can see that the independent variable is the DNA, the digestion of the DNA with the XBAL1 and APA1 enzymes. The dependent variable are the DNA fragment weights, and the control was the undigested plasmids. Our results were that the sample ID SP0 was confirmed to be was confirmed to contain the PRS413 plasmid, and that SP1 and SP3 were containing either the MSH2 plasmid or the MSH2 mutated plasmid. Although differentiating between those two plasmids containing the MSH2 gene would have to be found through PCR. So then following the DNA amplification using PCR, a restriction enzyme was used to digest the products, and the goal was to determine which sample ID contained the MSH2 insert. The SP3 was ultimately determined to be the sample ID containing the PMSH2 prime, the plasmid containing the mutated MSH2, and then with this known, we can use this for the yeast transformation following. The result of the yeast transformation, on the left, we can see the control plate, and this one had no mutated plasmid in it. It just had the yeast, the mutated yeast. And on the left, we can see the growth of the yeast colonies. There were approximately 600 colonies found on the plate. The plate that has the yeast following transformation is the result of the yeast producing their own nutrients despite them being absent on the growth plate. And the MSH2 gene and the HIS3 gene found in the mutated MSH2 plasmid, as well as the TRP1 gene found in the yeast's own SH44 plasmid, allowed for the growth in the absence of histidine, tryptophan, and uracil that was not found in the growth plate. Then on the left, in contrast to this, the control contains the untransformated yeast, like I've already said, and that was not able to produce its own histidine and other essential nutrients because it did not have the MSH2 plasmid nor MSH2 gene due to its mutated state. And therefore growth could not occur. That the independent variable was the transformed yeast. The dependent variable was the yeast growth that we see on the plate on the right, and that the control was the plate on the left, which didn't have the essential nutrients, but it did have the mutated yeast. All right. So next we have the results of the FOA. The objective here was to measure and compare the mutation rate of yeast carrying all three plasmids using the FOA assay. The independent variable here is the FOA, and the control is the plate lacking histidine, tryptophan, and uracil, and the dependent variable is the mutation rate. On the left, we can see the image of the plate that did not have the FOA on it, and on the right is the image of the plate that did have FOA. All three plasmids are plated on both of these images. However, as the columns continue, the concentration is decreasing. So you can see this more on the right image here. The first column, it's hard to tell the difference between the growth there, but as we use different concentrations, we were more accurately able to compare mutation rates. So the yeast growth on the plate lacking the histidine, tryptophan, and uracil, obviously seen on the left, is evident. We were able to look and see that the SNP, our specific SNP, increased, and the mutation rate increases the mutation rate in yeast, and that's that middle row there, the SP3. That's the sample ID on the right. Yeah, that middle column, that middle row there is what we're looking at, and it's in between, the results are in between that of SP0 and SP1, and so ultimately, we see that the mutation rate is increased due to the growth of the yeast transformed with the MSH2 mutated plasmid on the FOA plate. We can assume that the MMR mechanism is not correcting the frame shift mutation, and this would mean that the GT16.5 URA3 gene, which Chloe talked about in the methods, is not being properly expressed. and that the 5-fluorocyl is not being produced by the yeast, which would cause ultimate death. However, there is uracil on the plate. So through this mechanism that creates uracil for the cell is gone, there's still going to be growth due to that uracil being on the plate. And then ultimately, we found that the result of the MSH2 mutation is neither a fully functioning nor fully inactive MMR.
So the results indicate that the mutation may not be explicitly connected with MSH2 alone, but rather with the entire interactions of the MMR mechanisms. The SNP G704S does increase the mutation rate in yeast, which may correlate to a higher probability of developing HNPCC in humans. Overall, this research is imperative to the clinical surveillance of HNPCC, and it can help determine which SNPs put patients at risk of developing cancer. Specifically, this research could be helpful in detecting and even preventing, possibly, specific SNPs that put patients at risk of getting this cancer. And that concludes our presentation. And next, we just have our references. Any questions?